It's a pleasure to be here. I want to acknowledge my co-authors. Alicia Pedro Sampaio is the longest fellow with me, 4.5 years. And the first 4 projects in rabbits on losartan, she led those projects. And my 2 current fellows, Barbara and Valeria. And then Siju just recently went to the dark side. He's working in retina right now. And Marconi was one of my prior fellows who's very prominent in Brazil. I want to acknowledge that the Cleveland Clinic and I have submitted 2 patents on topical losartan. And we're very close to licensing to a company in Greece for international rights, a company that already has 6 drugs in the US and seemed like the best match for us. I've been interested in corneal scarring for a long time. This is actually my 2nd novel. I've written 4 novels, and 3 of them are the Stone Waverly trilogy. They're action-adventure novels. And this is one of the main characters of my 2nd novel in the Stone Waverly trilogy. Tanya was actually blinded by sulfur mustard gas during the Iran-Iraq war in northern Iraq in 1988, and that has a major part in the story. My lab, when I was trained in molecular biology and biochemistry before I went to medical school. And when I was leaving my fellowship in cornea at LSU, I knew I wanted to start my own lab, and I decided that I was going to focus on growth factors and growth factor receptors and cytokines in the cornea, because at that point in 1989, the only work published on any of that was Gospaderovich's early pioneering work on FGF1 and TGF-beta in corneal endothelial cells. And literally over the last, so we worked on HGF and KGF in the cornea, TGF-betas, of course, PDGF, IL-1, alpha, IL-1 beta, you name it, we've worked on them. And a lot of companies have approached me over the year with this and that new drug that they thought would treat corneal fibrosis. So we've actually studied dozens of them with industry support. None of them worked. So I'd like to start this lecture by saying I worked for 30 years on finding a medication for corneal scarring, and it was in my medicine cabinet all along. Losartan for 40 years. And a really important part of this whole story is initially when we started this work at the beginning of the pandemic, we couldn't get pure losartan powder. We could only get the tablets. So we crushed those. I didn't know much about how tablets were formulated. We crushed those, did solubility studies. It was soluble to about 1 milligram per ml, the material from the pills. So I decided in the first study, we'd start somewhere in the middle, like 0.4 milligrams per ml. Only later did we discover that the incipients that are in pills are 75% of the pills. That's what was precipitating. When we finally got pure losartan, we found out that it was soluble to over 400 milligrams per ml in BSS. It becomes like Helon, very viscous when you get up at that higher concentration. If I'd have gotten that from the very beginning, I'm almost certain that I would have tried concentrations like 100, 50, 10, which would have all been toxic. And this project would have never gone anywhere. But it turns out that in the first study, we were using 0.1 milligram per ml. We thought we were using 0.4, but it was 75% incipients. In the second study, we were using 0.2. And they worked. But I'll get to the rest of the story, how we got to 0.8, which we're currently using as I go along. So I credit the pandemic for this discovery, because if I'd have gotten pure losartan, I don't think it would have ever happened. My first interest in losartan was when I read this paper that was published in Science in 2011, where oral losartan attenuated TGF-beta induced fibrosis in a mouse aortic aneurysm model. I thought, hey, that's pretty interesting, you know, but I was fully funded at that point, and I just put that aside, that idea, until I started worrying about NIH eccentricities of funding, and decided, you know, I had better apply for another grant too. And I applied for a DoD grant, and that's what this was focused on. So we didn't start working on this till about 6 years ago. My original interest in scarring fibrosis was derived from the interesting cases of PRK that we did in the very beginning, where if you did 9 diopter PRKs on patients, humans, 95% of them would do great. 5 or 6% would develop late haze, corneal fibrosis. But if you did the same thing on 4.5 diopter corrections in humans, none of them got haze. And it was like, what's the difference? You know, it's a little less energy and stuff, but it just didn't seem to make sense. We found out that 9 diopter PRK in rabbits, fortunately, produces the haze in 100% of rabbits, because if it produced it in only 5%, you could never afford to do the research. Especially our rabbits now cost us over $400. I don't know what yours cost, but it's gotten absurd. So this was the beginning of my interest. And the same fibrosis is present in myofibroblasts account for probably 98% of corneal scars. So a buttonhole LASIK flap you see here, or the edge of a LASIK flap, or the donor-recipient interface in a penetrating keratoplasty, or any microbial ulcer, this happens to be a bacterial one, those scars that develop are all mediated by myofibroblasts. Probably the most important thing that led to this was when Andre Torricelli was in my lab. We looked at the electron microscopic level at the difference between these corneas that got 9 diopter PRK and 4.5 diopter PRK. And the difference was pretty obvious. This is 1 month after treatment. In the top panel here, you see the, see if I can get this working. The normal cornea, the lamina lucida, lamina densa that you all learned about in your early training. At 4.5 diopter PRK at 1 month, it's regenerated completely normally. But in the 9 diopter PRK rabbit corneas, you could not see any evidence at 1 month of a basement membrane. We now know there's components there. They're just not organized properly, and they're lacking in components made by corneal fibroblasts, which are collagen 4 and perlecan. So this discovery prompted us to start looking more at the basement membrane. We should have known that it had something to do with the basement membrane just clinically, because when PRK patients got haze, it was always immediately beneath the epithelium. So that should have been our clue that there's a reason they want to be there. And the reason is they're getting TGF-beta from the epithelium and the tear film if there's a defective basement membrane. Two of the mechanisms we think account for this is, number one, stromal surface roughness. We know when you do a 9 diopter PRK, you're much more likely to have surface irregularity, which mechanically kind of impedes basement membrane regeneration. But probably more importantly is our work where we've shown that the epithelial basement membrane actually should be called the epithelial stromal basement membrane, because the stroma makes several of the most important components. The epithelium starts by producing the laminin layer, and then the corneal fibroblasts come in and supply most of the collagen 4 and perlecan components. Nitrogen is also produced by both. So we think that when you, if you have insufficient keratocytes, like you wipe them all out from a high correction surgery, you don't get enough corneal fibroblasts regenerate to help the epithelium regenerate that epithelial basement membrane. There may be other causes too that we haven't landed upon yet, but those are two of them that we think are at work. One of our early models was when Gustavo Marino was in my lab. We did a Pseudomonas ulcer study where we infected corneas with Pseudomonas. And then the next day started antibiotics, sterilized them so they were no longer infected. And by 1 month, you can see the red here is staining for alpha smooth muscle actin. In this particular cornea, 95% of the cornea was affected, sparing the posterior and the endothelium, you could see. Some of the corneas it went all the way through, as Pseudomonas sometimes does. And some of those corneas were like this one. This is 2 months though. I'm sure at 1 month, if we'd have taken this cornea, the whole cornea would have been filled with alpha smooth muscle actin myofibroblast. But by 2 months, the epithelial basement membrane had regenerated, as you see in the bottom panel. And therefore, the most anterior myofibroblasts, most of them underwent apoptosis because they were no longer getting the TGF beta 1 and 2 that they needed from the epithelium and the tear film. Whereas the endothelial damage was not regenerated by then. It takes 6 months to regenerate the rabbit endothelium, and it's probably forever in humans. So a lot of these we saw had, if it went all the way through, they still had posterior fibrosis at 2 months, 3 months, and 4 months. After it turns out by 6 months, it can disappear. The endothelium in the rabbit can totally regenerate Descemet's membrane, and all of the myofibroblasts then disappear. When we saw that, we were wondering, well, what is it? Is it the endothelium or is it Descemet's membrane? That's the important structure because we were removing both. So we designed experiments where we either damage the endothelium with a smooth cannula, like you're seeing in the A panel here. If we do that, the next day the cornea is edematous. But in the rabbit, within 2 weeks to 4 weeks, it's back to normal. There's no fibrosis that appears. But if you do desmetorrhexis and remove the Descemet's and endothelium over 8 millimeters, then you get this tremendous fibrosis response. And looking at these with immunohistochemistry, the green is keratocan, which of course, some people here, that's their favorite molecule in the world. And this is an antibody that's produced by Winston cow. It's not commercially available. And now Winston's retired. And we just got his last file, and you can't have any of it. Anyway, I'm holding on to that till my retirement. Once I retire in 2025, you can have the rest of it. But anyway, I kept telling him he needed to market that.
Yeah.
There would be a lot of people that would like to have that antibody. Works in rabbits, works in humans. But in the ones that had desmetorrhexis, here stained, both sides were double stained for alpha smooth muscle actin in the red and keratocan in the green. And you see the layer of fibrosis in the posterior cornea of all these corneas that had desmetorrhexis. So it really was Descemet's membrane, which we suspected, but this really demonstrated it. Well, about this time, as I told people, I had I knew the source of TGF beta for that posterior cornea was the aqueous humor. Wayne Strayline showed that many years ago, that it's full of TGF beta 1 and TGF beta 2. So we decided when I wrote my first DoD grant, I decided that that's what I was going to focus on, and that was going to be the first model I was going to use. Because if I can go backwards, you can imagine if I use corneas like this one on the right, the epithelium and epithelial basement membrane are normal. So if it would work on this model, it could work on anything. So that was the first experiment in the DoD grant, and this was the results. We, of course, tried oral losartan at a massive dose, equivalent to a human getting 400 milligrams a day, which we don't do that to humans. And the veterinarians had to approve what dose we could go to. Didn't notice any untoward effects in them, but a very high oral dose had no effect in the top panel here on the unwounded cornea, oral or topical. But if you did desmetorrhexis, the entire cornea becomes fibrotic in the posterior half of it, if we were to look at a slit lamp exam. And if you use the oral, there was no effect. Oral losartan by itself this is at 1 month of treatment. But if you use the topical losartan, you can see the massive clearing in the peripheral cornea which I'm sure would have continued if we'd gone on to 4 or 6 months, but we can't afford to do that. We stop everything by 1 month or six weeks. In the oral plus topical, there was no added benefit of the oral. You just got the topical effect. So it turns out we thought we were using 0.4 milligram per ml. Then I had to send in a correction when I discovered that pills are 75% garbage, and we were really using only 0.1. In the second study, since we didn't see any toxicity in this one, the second study we did, I'll come back to that. We went up to 0.8, we thought, but we really were only using 0.2. But the world saw 0.8 in that publication, and clinicians all over the world, especially in Brazil, started using that in patients and didn't notice any problems, and it worked. And that's how it became the adopted concentration until I'm going to show you some reasons why for some patients that's not the right concentration. Another thing we discovered in this is that the corneal fibroblasts, when you injure the cornea, the collagen 4 isn't just in the basement membranes, decimase and the epithelial basement membrane. Corneal fibroblasts make all kinds of collagen 4 throughout the cornea, and it binds directly TGF beta. And I think that's a way for the cornea to try to downregulate the TGF beta response. We published this in Matrix Biology and suggested that it's probably present in other organs too. And several people have found it in liver and lung and other places. So this proves that the losartan was getting all the way through, because how could it suppress the production of the TGF beta? How could it suppress the collagen 4 production at the very posterior cornea unless it was getting back there? Going back and forth. Then the next study we did was alkali burns. And we discovered in the alkali burn studies that the combination of steroid and the losartan worked best, was the most reproducible in each eye. And so in inflammatory conditions, we're now recommending topical losartan with the steroids. So that would be things like microbial ulcers. Of course, you're going to give them the antibiotics too. But anything that has a lot of inflammation, losartan has no anti-inflammatory effect, so you use them in combination. And this is just showing why you couldn't use the acetate by itself. Some of those looked just as good, like this one down here. We only had myofibroblasts at 1 month in the very posterior cornea, but another one, there was still fibrosis throughout the stroma in the prednisone acetate alone group. But all of the corneas in the prednisone acetate plus losartan only had a little bit of remaining fibrosis in the very posterior cornea. If we'd have gone on with treatment, I think that would have been eliminated with time, as we're seeing in patients treated for longer. And this is just showing, if we look at total alpha smooth muscle actin opacity with image J technology in the central cornea, that in the vehicle alone, you get a lot of myofibroblasts, and losartan knocks that down. Predacetate can do it in some corneas, but not in others. But if you use them together, you get a really tight spread of the control of the fibrosis. Well, so the boys from Brazil, but there's also girls from Brazil, all saw these publications and immediately started using them on their patients, like Renato Ambrosio, Marconi Santiago, Rodrigo de Oliveira. And this was the very first one. This was a LASIK patient that was referred to Renato, that had LASIK with a lost cap. The cap was replaced. The patient got severe diffuse lamellar keratitis inflammation along the lamellar interface and ended up with scarring that limited the best vision on the left to 20/80 in that eye, best corrected. He started 0.8 milligrams per ml with pure losartan, not the pills. So it was really 0.8. Patient did fine. Notice the epithelium was already intact in this eye, which if it hadn't been, he would have probably gotten a different result, as I'll show later. And by 5 months, the clarity was completely restored in that patient. Some other examples, this is a patient that had radiokeratotomy like a decade ago, was fine for like 9 years, and then started developing fibrosis in one of the incisions that was progressing into the visual axis and decreasing his vision. And this is after only 15 days of the losartan. You'd never see a scar change that quickly unless something is affecting it. And he was so good that after a month, he left, and he hasn't been back yet. So it hasn't recurred apparently, but I predict he'll be back because I don't think he was on it long enough for reasons I'll talk about later. Then this was a clinician, Danielle Medros in Brazil. This is a acanthamoeba scar. The left is a year after treatment, so there was no longer any infection, but she was left with this scar. And this is the picture at 4 months after treatment. And she has continued on the losartan. I think she's about 9 months now. And Danielle has promised me the photos, but I haven't, but she told me verbally that she's a lot better. I told her to keep going for at least 6 months after all the scarring is gone. Otherwise, the basement membrane is probably not repaired and it will probably recur. Another Staph aureus ulcer that Rodrigo treated, pre-losartan treatment, the epithelium was intact by now. Then at 1 month and 4 months of treatment. And then any of you get on Keratin? Okay, I've tried to get on there twice, and I put my, and I never get on. I don't know why. But somebody sent me these pictures that Whitney Potter had put on Keratin that from Oklahoma. This is a EKC case, not the acute stage where they're responsive to steroid, but after a year of being on the steroid, they finally went off the steroid and ended up with these permanent scars, treated with losartan. And I think this is 3 weeks treatment. I told him, go keep it going for 6 months. He did. The patient's off of it now for 4 months and has not recurred. So this is not the inflammatory steroid responsive stage. This is after they're really scarred. And then one of the most interesting ones that I just recently got day before yesterday, this is also from Renato Ambrosio and Marcella Salome, who's also one of my prior fellows. And they only sent me the OCT so far, but you can see the bottom is she had zoster in 2020. So she had it for 4 years, and they decided not to transplant that eye, just save it as a spare. I don't know how many times I've heard that from patients. Now he told me just to save it as a spare. And the top panel is after only 5 weeks of treatment. She's continuing, and her vision's already dramatically better. That's one of the interesting things about treating patients with losartan. I can't treat them myself, according to the Cleveland Clinic policy, since I'm the inventor. So I have to refer them to my colleagues, but they keep me posted on what's going on. But I get them all. I go, yeah, you'd like to benefit from it, and I send them to the other people, which I don't feel great about, but that's Cleveland Clinic rules. So she's continuing, and the idea is to continue until she has complete clarity. She's down to 2050 now from 2200, and let it completely clear and then go 6 more months is what I'm recommending at this point. Losartan works by interfering with activation of ERK, which is a really interesting signal transduction molecule in the non-canonical pathway, TGF beta. And you see down here where it works. Now, ERK, once it's activated, anybody getting ready to write some new grants, this is a prime area for it, because ERK controls about 150 different genes, and almost nothing's known about it, what those genes do. We know that they control viability of myofibroblasts, their collagen production, and lots of other things, but the individual genes are not well understood. So I just wrote a review on this. It's an experimental eye research. And I pointed out, if I was starting a lab right now, I would focus on those molecules because so little is known about them. but it blocks the activation of ERK. And ERK, once that signaling is withdrawn, the myofibroblast undergoes apoptosis. So it's a two-phase process of treating with losartan. The actual elimination of the myofibroblast occurs really quickly, within a week or two. But the basement membrane has not been regenerated yet. So if you stop it immediately after you see the clearing, there's a good chance that you're going to get the basement membrane not regenerated, and the TGF-beta 1 and 2 will come back through, and the haze will regenerate itself. So that's why we're recommending for 6 months once you get full clearing. There's no side effects, by the way. There's no stinging with the 0.8 milligrams per ml or 0.2 milligram per ml. None of the things that, for example, I was involved in Restasis development, and that was always the worst problem, was it stung in most patients. This one doesn't. So that second phase, where the myofibroblasts are gone, but the corneal fibroblasts have to come in and regenerate the basement membrane and reorganize all those disordered collagens the myofibroblasts produce. That's the longer phase. But you got to keep the losartan going during that phase, or it'll just recur. This does point out the important concept that topical losartan can be used prophylactically. So you could use it in a patient with a bad pseudomonas, so the first day you see him, you treat the pseudomonas, but you could also start losartan in those patients, or you can treat them therapeutically once the scar is already, in most humans, that's how they've been treated in the world so far. But you could use it early on in something you know is going to scar. To date, the longest duration is 3 years, but I'm hoping we go out 10 years. I think some of those that have been there 10 years are going to respond, because the myofibroblasts have to continue being there, maintaining the scar. It doesn't maintain itself. So that led us to the question, can we use higher than that? Okay, there was one Brazilian pharmacy that for 3 weeks made a mistake and was giving 8 milligrams per ml. The epithelium was intact. They didn't see a problem. So a bunch of Brazilians started using, oh, let's use 8 milligrams per ml. Well, we decided we better go look at that. So we decided to use an alkali burn model that I published, a 0.75 in AOH on a circular sponge for 15 seconds, just produces death of keratocytes all the way through the cornea. And in the anterior cornea, you're seeing the anterior fibrosis, but it also damages some of the endothelial cells. So you also get some posterior fibrosis in this model. And we decided to look at 0.8, which everybody was using 6 times a day for 30 days, 8, which this Brazilian was using, fortunately, in corneas that had their epithelium intact, or they would have said losartan doesn't work, it causes persistent epithelial defects. And then the 40. The 40 was so traumatic to the rabbits that we had to stop at 9 days. When we put the drop in, they would run around the cage screaming. And I finally said, we're not doing that anymore. And so we terminated that part of the experiment. So don't do that in humans. It will sting if you go up that high a concentration. But even the 8 milligram per ml, and this was started at the time of the injury, 5 of the 6 got persistent epithelial defects in the 8 milligram per ml. The rabbits didn't seem traumatized by the drops, but the corneas didn't heal right. And even in one of the 0.8, we got a persistent epithelial defect. Now, you can get those just with a alkali burn. So we don't know for sure that it was the losartan. But since the original study, it worked at 0.2 milligrams per ml in the decimeter rexus model, we're now advising people that there is obviously some toxicity with higher concentrations. So if there's an epithelial defect, we're recommending using 0.2 milligram per ml until the epithelium heals, and then going to 0.8, or if the epithelium's already healed, use 0.8, 6 times a day. Some people, of course, are trying 4 times a day. And I said, oh, it'll probably work in some of those patients, but some it won't. You'll probably have a higher failure rate at 4 times a day, maybe because some patients, if you tell them 4 times a day, they may only get it in 2 or 3 times a day. Hopefully, if you tell them 6 times a day, some get it in 6 times a day. Maybe those are the ones that are clearing so rapidly. And that's all going to have to be worked out in clinical trials. This is showing some of the immunohistochemistry from that last experiment. And this is all the 0.8 milligram per ml group, 6 corneas, staining for, it's a combined SMA and LAMA5 and Vimentin. And basically what you're seeing here is that by 1 month, there are no myofibroblasts anymore. So they've already been eliminated, but the scarring is not gone and the basement membrane is not regenerated. So you've got to go longer or it's going to likely recur. And a similar thing in the 8 milligrams per day, except there were more myofibroblasts present. In this case, they're the green ones. I'm sorry, the red ones. Can you see those?
Well,
Maybe on that screen you can see them better.
Yeah.
But the 40, remember, this was only 9 times a day, was even worse. The higher you went in the concentration, you started having the opposite effect, probably because it was injuring the epithelium. If you plot this out by determining alpha smooth muscle actin cells per corneal section, as you went to 8 and then 40, you actually got an increase in myofibroblasts. So don't use the higher concentrations. So that has led us to this kind of way to use it, in our opinion. Any patient that has an epithelial defect, which is a traumatic or a chemical burn, or PRK and PPK, or a microbial infection, or cross-linking, if they have an epithelial defect, only use 0.2 milligrams per ml 6 times a day until the epithelium heals. And then you can go to the 0.8 milligrams per ml. I don't know if the two will really, you might be able to just stay on 0.2. Somebody needs to do that experiment. And I'm sure it will eventually be done. So what kind of things can you use it on? Almost everything that is scarring, other than things like the transient haze after PRK, which is corneal fibroblasts, or the transient haze after cross-linking, which is corneal fibroblasts. You don't need it there anyway. They're going to go away on their own, but it doesn't affect the corneal fibroblasts. But thermal and chemical burns, any type of infectious corneal process, it's been used in all of these, and I've seen the cases where it's worked. Herpes simplex, recurrent keratitis, you can use it. You want to keep the antivirals going, but they're using it, say, acyclovir. If the ulcer's gone, the epithelium is healed, they're using like twice a day acyclovir and then the losartan to get rid of the scarring. Or shingles, some of my favorite cases have been herpes zoster's cases. Corneal lacerations, we just had a paper published in Cornea, and it shows that you can create lacerations and the losartan doesn't inhibit their closure. So it's the corneal fibroblasts doing that, not the myofibroblasts. So you can use the losartan, it decreases the opacity, but you don't have to worry that it's going to cause the wound to open up. It didn't do that. So that was a good thing because a lot of people would like to use these on corneal lacerations. Late haze PRK. I know of 3 or 4 cases with that now. And persistent epithelial defects is an interesting one because we know that persistent epithelial defects all have myofibroblasts in the bed by a week of a persistent epithelial defect. And NGF is currently the only treatment if your patient has $100,000 to spare for a 6-week course. But I'm interested in whether losartan could benefit these patients because it definitely can inhibit the myofibroblasts. Myofibroblasts inhibit the nerves regenerating. So as long as the myofibroblasts are there, it's probably going to perpetuate the persistent epithelial defect. So either in combination with an NGF or maybe by itself, see, when people start trying it, it might have an effect by eliminating the myofibroblasts, allowing the corneal fibroblasts to come in to not only regenerate the basement membrane, but also those growth factors control the proliferation and motility of the epithelium. So there's reasons to think it might be useful. I don't know of anybody that's done that study. You might do that here. So, but it won't, there's other possibilities for it. All of the conjunctival fibrotic diseases, if it gets all the way through the cornea, it's likely going to get through the conjunctiva. Victor Perez started when Victor Perez was at Duke, and he just left Duke about 3 months ago. He started studies at Duke, and people were improving. And then he had to leave to go to Miami. He doesn't know if they're continuing those patients at Duke or not, but he started the new groups of patients at Miami, and he thinks it's working. So I don't see any reason why not. These all have fibrosis under the conjunctiva, and since the losartan goes all the way through the cornea, I don't see why it won't work in these. The other one I was trying to get one of my glaucoma colleagues to try is that scarring blebs and filtering surgery, that's all caused by myofibroblasts. And also encapsulation of the tube shunts, they've all shown that's myofibroblasts too. So I had no luck getting one of our clinicians at Cleveland Clinic. So I told Renato about it. And one of his glaucoma people has now organized a study with Wills and Bascom Palmer and is trying this. So we'll find out whether it works for that. Another interesting possibility, and I have no idea whether this is going to work, but the transforming growth factor beta induced corneal dystrophies, TGF beta is involved in the production of that matrix. So could the losartan affect that? And is it safe to use for someone's whole life? Or would this be the type of thing like when we do a PTK, we're forced to do a PTK or a transplant, they recur. Could you then start using it and prevent recurrence in the either PTK cornea or the graft? And Guillermo Rocha at McGill has started doing this, has a huge group of these patients, and he's started doing that in the last few months, and I haven't heard how that's working, but that is a possibility. So this just reiterates that. When would you do it? Could you use it in a kid? That's going to be risky, I think, to use it from that early age before you try it in some of the adults that had PTK or a transplant. And then, are there any retina people in here? No? Oh, there is one. Okay. Well, I've tried to get people in my department interested in this too, because we are a retina department at its heart, and I can't get them interested. But I do think some of the intraocular fibrotic diseases that you could use losartan, not topically, but with these extended release devices. And there's rabbit models of PVR. And so I always encourage people, I don't know of anybody trying it yet, but I think it's really worth a shot as a treatment for some of the intraocular fibrotic disorders. In the patent, we covered all of the known ARBs. There's, I think, 9 of them, but only losartan has good solubility and balanced salt solution. The other ones are either very little soluble or totally insoluble. So you'd need some vehicle like Restasis for cyclosporin has. So we've stuck with the BSS. As you saw, it penetrates the intact corneal epithelium and epithelial basement membrane. There's no stinging or other signs of ocular toxicity that we've seen. If you stick to the 0.8 or especially 0.2, because those were done with epithelial defects. And then I'd just like to acknowledge all of my fellows that have done all this work all these years. The ones in the yellow are winners of the Troutman Award that's given each year for the best paper in Journal of Refractive Surgery. And then Rodrigo just won the George Waring Award recently. So I couldn't have done this work without him. And of course, NIH and DOD.